Well, Alison, sorry I couldn't be there. I know we had to cancel for a personal reason at the last minute, but glad that we have a chance to update on the work that we've been doing ever since I met Paula and you several years ago. But before I get started, I do want to acknowledge those that do the work. I tend to forget about that. And that is Justin Percival and Ralph Schmidt, as well as many people at the gene therapy program that provide core services. Next. So I'm going to describe the paradigm of gene therapy translational research in the context of animal models to human. Next. We begin with the study of the therapeutic in an animal model that until recently has primarily been mouse studies, in which we study gene correction or transgene toxicity. And as we heard in the previous talk, there's a potential for other larger animal models as well to determine whether we can correct the disease. But then we need to move into a larger animal next, which we use the non-human primate or monkeys to study safety and dose scaling. And below just shows the scale of brains from a mouse to a human, which would also indicate some of the challenges of modeling scaling from a mouse. And then finally, if we put together a sufficient data package, we moved to humans next, in which we study safety and possibly biomarker readout. So we've done this approximately 15 times in different gene therapy programs, from mouse to monkeys to humans. And we hope to leverage that experience for Angelman syndrome. Next. The real challenge, however, is getting enough gene distributed into the globally throughout the brain in order to achieve a therapeutic effect. And I want to describe how the route of administration may be important. The initial approach was to use an intravenous delivery, but this requires that our gene delivery vehicle, which is we call a vector, crosses the blood-brain barrier. Next. Another approach that was developed for certain neurologic diseases is neurosurgical, and where you inject the gene vector directly into the brain through multiple different injection sites. It isn't very efficient and not relevant for Angelman. Next. We instead are considering a different approach to inject the vector into the fluid that bathes the spinal cord in the brain, either through the lumbar area or next, into the area closer to the brain where the CSF resides, and that is into the ventricle or into the cisterna magna. Next. This summarizes the tally of gene therapy studies that are in the clinic using AAV vectors, delivering vectors in different routes of administration. It turns out that the majority of the different approaches utilize an injection into this cavity called the cisterna magna, that's at the bottom of the neck, or directly into the brain. So there is an emerging experience. Next. I'm just going to summarize probably the most celebrated and the most transformative application of AAV vectors for a neurologic disease. And it was based on a vector that we discovered about 20 years ago called AAV9 that, when injected into the blood, crosses the blood-brain barrier to a certain extent. Next. And this is a picture of an experiment done by Ron Crystal where he labeled an AAV9 vector and injected it into the blood in a monkey. And then you follow the distribution of that vector over time, 24 hours and later time points. And you can see that the vector primarily distributes to the lung and the spleen, not into the brain, but a little bit into the spinal cord. So while it penetrates the blood-brain barrier some, it's somewhat marginal. Next. How does this translate to actual transfer of the gene and expression of the gene? And this is shown in a dog model using an intravenous delivery. Next. These are histological sections of a dog that received a vector intravenously. And we stain the tissues for the presence of cells that express the gene. It turns the cells red, and you can see very little red around the brain, the frontal cortex, a little bit around the cerebellum, but it turns out certain cells of the spinal cord are accessible to the vector, and these are the motor neurons. Next slide, please. This was applied an AAV9 vector delivery intravenously to the disease spinal muscular atrophy type 1, which is a neurodegenerative disease in which children are diagnosed before the 6 months of age, and it leads to a fairly accelerated course in which they die from their disease within 12 to 18 months. Next. The root of the disease happens to be in these neurons, motor neurons, that emanate from the spinal cord into the muscle. And due to loss of a gene function, these neurons degenerate, and it leads to loss of muscle function. Next. So it turns out those cells are accessible by an AAV9 vector intravenously, and a drug was developed by a group at Nationwide Children's, licensed to a company of Exis, and then acquired by Novartis, called Zolgensma, that has now changed the landscape for families and patients born with SMA type 1, in which they're now living and actually gaining milestones. So this gives us hope that a gene therapy approach for a neurologic disease may indeed be successful. Next slide, please. But for Angelman syndrome, we need to introduce the gene into cells more than just the spinal cord. We need to distribute the gene throughout the brain. And this is, it was because of this challenge that a graduate student in my lab, Christian Hinderer, began to explore other ways to deliver vector into the fluid that bathes the brain and the spinal cord, the CSF. So one approach is to inject the vector into the CSF down lower in the back, what's called a lumbar puncture. Next. Next, please. Okay. The other, no, you can go back one, please. The other approach, which was illustrated in the pig experiment in the previous talk, is to inject a vector right below the occipital bone of the brain in what's called the cisterna magna that is closer to the brain. So we asked the question, in monkeys, which is the best way to get vector to the brain? Next slide, please. So in this experiment, we injected an AAV9 vector into the lumbar space of monkeys or into the cisterna magna and wanted to know where did the vector go and which would be better for distribution of vector into the brain. Next. This shows on the y-axis the presence of the vector genome in various tissues of monkeys that receive vector, either in blue, a lumbar infusion, or red, a cisterna magna. And let's look over to the left. Next. And I pointed out the difference between vector delivered into the lumbar space versus the cisterna magna in terms of deposition of vector into the brain and the spinal cord. And you can see a much better delivery of vector to the brain of monkeys when delivered via the cisterna magna. And on the right side of this slide, Ron Crystal has taken an AAV9 vector and injected into the cisterna magna of monkeys and followed them over time, visually following the distribution of the vector along the brain, the spinal cord, and outside the central nervous system. And you can see, as opposed to the intravenous experiment I showed in previous slides, that a lot of the label that can be imaged in these monkeys by 24 hours is bathing the brain, and then it slowly descends down the spinal cord. So it was really based on these data that Christian Hinderer had developed that as we progress forward with neuropathic diseases, such as Angelman syndrome, that we preferred a route of delivery that involved the Cisterna magna. Next slide, please. So I also would like to share with you our experiences, extensive experience in hundreds of monkeys who have received vector via this route about potential safety concerns or toxicity. It's been incredibly well tolerated in monkeys, and now this has been introduced into a number of clinical trials. But there is one consistent finding in some, many, but not all vectors, which is toxicity of a nerve that is called the spinal nerve that is present along the spinal cord and projects to the peripheral nerve in an anatomical space called the dorsal root ganglion. And this was discovered from senior scientists in my lab, Juliette Hordau. And what we found is when we inject vector into the cerebral spinal fluid in monkeys, depending on the circumstance, that we see inflammation of these nerves. Next. As shown here in a histological section, the nerve bodies begin to degenerate. And when they degenerate, they're axons that project peripherally and centrally. Next. Also begin to look damaged. Now, we've studied this in hundreds of monkeys, and it's been a consistent finding. But the question is, does it have any clinical correlation? Next. And the only time that this has demonstrated any clinical manifestations is when we use a protein that is very foreign to the host, in which there is an superimposed immune response. But when that happens, they develop discoordination and ataxia and tremors. Since vectors delivered into the cerebral spinal fluid are now in a number of clinical trials, the question was, is there any evidence clinically in patients, which ultimately is the most important model to ask the question. And fortunately, it seems to have largely been without symptoms. However, there was a case study of a young man with an accelerated form of ALS who received an AAV9 vector into the lumbar space that soon thereafter developed what was called neuropathic pain, like a diabetic would have, that required analgesia that eventually resolved. But when we think about delivering a vector into the cerebral spinal fluid in any way for Angelman, we have to pay particular attention to this kind of safety or pathology. Next slide, please. So Juliette has gone on and actually developed mitigating strategies to prevent this. But I just wanted to point out what we think the mechanism is, because then we can focus our studies in Angelman syndrome based on a model. This is a picture of the sensory neuron that we believe is damaged by high doses of vector. It's called the dorsal root ganglion. Next slide. Next. And so when a vector is delivered into the cerebral spinal fluid, it is actively taken up by these cells and generally is okay. But if the transgene itself has any toxicity, the protein that is expressed, it could overload the internal system of these neurons and could lead to degeneration. So what we found is that the presence of any safety issues is really a function of the transgene. Some are less and some are more. So it was really important for this program to determine what we see in non-human primates that would be delivered an AAV vector expressing UBE3A. And I'll talk about that in a minute. Next slide. So when I thought about a strategy after I met with Allison and Paula about gene therapy for Angelman syndrome, it sounded complicated genetically, and got to meet Art Beaudet and learn more about the disease. But what was really fascinating about the complexity of the genetics is that complexity actually yielded a number of novel approaches so that you could pursue parallel paths that wouldn't be available in other monogenic disorders. And so as Allison had noted, we have four actually now, separate programs approaching this problem using different strategies. And when I think about what our goal is in the lab, I think about having as many shots on goal as we possibly can, hoping that one of them would make it. But the other part of our work in the preclinical space is to make sure that when we take a shot on goal, that we score. And so you don't want to be taking shots that are unlikely to score. And that is really the basis of preclinical data to try to mitigate the risk of failure or any potential toxicity. So I say have multiple shots, but have these shots on a goal that's close and there's no goalie or it's open. Not sure that's the way it'll play out, but that's the way we think about it. Next slide. So the different approaches for treating Angelman syndrome you've heard about. One is, and it's all based on, if we go to the next slide, a mutation on the maternal allele in the inactivation of UBE3A on the paternal allele. And so as we've heard, and what you'll, in previous talks and what we hear from us, that one approach, next slide, is to use molecular approaches or genetic interventions to reduce the expression of the antisense RNA that suppresses the paternal allele. And we're going to describe two approaches that we're developing in the clinic, in preclinical models. And in addition to that, another approach is, next please, is not to try to modify this inactivation, but simply to add a normal version of UBE3A that otherwise has been deleted in a way in which that is not inactivated itself, and that's a gene replacement therapy using AAV. And we'll also describe two strategies that we're pursuing using a standard AAV gene therapy approach. Next slide, please. So, so it all begins with an animal model. We heard the exciting emerging data about a pig model, but all of our work was based on really a mountain of experience using the mouse model. And this just illustrates several different strategies that are used to evaluate the phenotype or the abnormalities in these mouse. And it was really based on work done by others. And here we have in 4 different assays, the difference between a wild type, which is in black, and then the AS mouse model, which is in red, showing a change in the outcome of the rotorod, open field, marble bearing, and nest building as well. And so our first indication as to whether a gene therapy, a genome editing, or whatever would work is based on a readout in some of these particular assays to see if they become more like normal. Next slide. So in terms of standing up a gene therapy approach, we needed to recognize the fact that there are different isoforms of UBE3A, 1, 2, and 3. And the question is which gene, which form of UBE3A would you want to use? And we haven't decided yet which may be the best, or we may be able to develop an approach to express more than one. But our initial experiments used an AAV vector expressing UBE3A isoform 1 and 2, and next slide, evaluated the outcome of gene therapy when directly delivered, in this case, into the ventricle of mice, which is not the Cisterna magna, but close to it, and then subsequently analyze the animals for a variety of different tests, such as those that I just described. And the hope would be that we would achieve some reversal of the neurobehavioral abnormalities. Next slide. So this is a summary of our initial work, and I'm going to point to you in the table where we have isoform 1 and then isoform 2. And we've evaluated various different phenotypes, nest building, hypoactivity, longer stride length, impaired motor coordination at different doses, low and a high dose. And whenever there was a green, we showed an improvement. And whenever there was a red, there was actually a slight worsening. So far our data indicate that if we were to use only one isoform, it may be isoform 1. So we then moved to the next step, which was to take our best vector that demonstrated the best data in the mouse models and evaluated that vector in non-human primates. Next slide. So the non-human primates do not have Angelman syndrome, but why they're so helpful to us is they better simulate than any other species whether the host will mount an adverse event, a reaction to the vector or the treatment, and best simulates the distribution of the vector to the cells that we need it to distribute to. So we really believe non-human primates are a gate to moving into humans. So we began some pilot studies. Next slide. And this just shows in some animals that were delivered into Cisterna magna and our UB3A vector, the vector copy number as it's measured vector copies per cell, and one would indicate one vector copy per cell in a variety of tissues. So we were getting pretty good vector distribution like we had seen in many of our other programs. Next slide. Then we also evaluated those same tissues for RNA from the UB3A. And if this wasn't present, then this would really be a non-starter, but we were able to detect RNA derived from the vector in all the tissues. So the gene was there and there was some level of expression, but we didn't know how many cells had taken the gene up, although we had a lot of vector genomes, or what the level of expression was. Next slide. But what we were able to do is in these studies ask the question, will there be, or is there this toxicity of the spinal neurons? And this was an experiment that I'll never forget because it's the only model that we've ever evaluated ICM AAV vector where we had absolutely no evidence of pathology in the spinal nerves. So for whatever reason, and sometimes luck is on our side, expression of UB3A does not, in this context with these vectors, does not demonstrate the kind of toxicity we've seen in most other studies. Next slide. So the question is, and I think this is the question, are we getting enough distribution of the vector, and is it expressing enough in those cells to be therapeutic? And what we've seen and others have seen is there is a difference in the ability of distributing vector and expressing genes in cells when you compare mice, and this is in the context of a related program but we're generating similar data in AS, where we show in mice many green dots, which means a lot of the neurons have taken the gene up when injected into the cerebrospinal fluid. When we do the same experiment in monkeys next, what we show on the top are the number of neurons in a section of brain, but only a few of those can we detect the expression of the transgene using this approach which is called in situ hybridization. So this would suggest that only a few percent of the cells, or less than 1%, are actually expressing the gene. But we were concerned that the method that we use to detect expression may not be sensitive enough. So next, we conducted studies, rather laborious studies, where we took these brain tissues, we segregated out individual nuclei of neurons, and then probed those individual nuclei for the presence of DNA or the presence of RNA. And we were quite surprised, pleasantly surprised, that up to 60% of the cells actually have vector, and many, if not most of those cells, are expressing RNA, albeit at lower levels of RNA. So where we are now with respect to gene therapy for AS are data in monkeys that suggest that we get pretty efficient distribution of genome and expression of that genome, although it's apparently at low levels, escaping traditional methods of detection using tissues. And we've got to determine whether we think there's enough expression per cell to proceed. Next slide. However, we're pursuing a parallel approach with our collaborators at Amicus, and that really was based on the observation that of potentially generating a secreted version of UB3A, next, that could then be taken up by a cell, but then secreted from that cell, next, taken up by other cells, next, next, and then next, correct those cells. So we may be able to get more bang for our buck by using a limited number of cells expressing a secreted version of this protein. And if we could demonstrate this, then we actually may be there with respect to the level of gene transfer. Next. So we're working with Amicus on a collaborative program based on work that had been done by a number of other investigators in the field to modify UB3A so that it has a portion of it to be secreted by the cell and a portion of it to be taken up by the cell. Next. So stay tuned with that collaboration, but it's off and running. Next slide. So two final approaches to briefly tell you about, and that is the use of genome editing to permanently knock down the antisense RNA. Next. So in this approach, what we've done is we've used AAV vectors to introduce CRISPR-Cas9 at a portion of the gene proximal to UB3A to permanently inactivate the ATS. Next slide. And we've screened a number of molecules, they're called guide molecules, for those that had the greatest activity against the mouse version of this portion of the genome, and we found a number that were. And we published this results in a paper. Next slide. So we asked the question, if we injected an AAV vector into brains of the AS mouse model, can we edit, and this would be a one and done, edit that portion of the genome to suppress the ATS to reactivate the paternal gene? Next slide. And this shows a success. On the left is an analysis of the brains of the mice receiving the editing construct, where we show the appearance of a black band, which is the protein that was reactivated. And you can also see by measuring the tissues that we now have brown speckles that we didn't have before. And those are cells that now have been permanently reactivated from genome editing. Next slide. And this is just a higher magnification view. On the left is a wild type, and on the right is an edited knockout mouse. Wherever there's brown, we've successfully edited that gene, so it now is at expressing the previously inactivated gene. Next slide. So we asked the question, could this actually impact on the behavioral abnormalities of the mice? And what you're going to see is in black, the wild type mice, normal, treated with AAV, and then the knockout, which would be in blue versus red. So we'll compare blue versus red to see if it's better. Next. So you see that in editing in terms of the rotorod, it seems to be better. Red is better than blue and closer to black. Next. Nest building, you can see that red is approaching normal and significantly better than untreated. And next. Marble bearing, another readout, we show demonstration of some benefit. So next slide. So I'm just going to end on more preliminary work that has been funded by FAST. And again, the gene therapy work and the entirely funded by FAST. And this work, Allison really encouraged us to proceed. And it's really based on the pioneering work of the use of ASOs to use a mechanism of RNA interference or antisense oligos in a similar approach using RNA interference to prevent the expression of the ATS RNA. Next slide. The difference here would be that rather than repeatedly deliver the ASO, that we would deliver an AAV vector that would express an RNA that would constantly be present to inactivate the presence of the ATS and reactivate UB3A. Next slide. So we've focused on what's called a microRNA gene that can be expressed and produce RNA interfering molecules in a region that's in the ATS region, very close to the inactivated UB3A. Next slide. Ralph conducted a series of in vitro experiments to screen for microRNAs against different regions of the genome. Next. I think he presented that. And we found, based on in vitro studies, these are cells from a mouse model that is a great way to indicate activity, that there were some microRNA targets that we could get re-expression of the paternal gene. Next slide. So we then moved this into mice, and this is the status of the work. We could express high levels of these microRNAs. Next. And that this did lead to downregulation of the target region that we had directed it against, not completely, but at least partially. And then the question is, would this lead to reactivation? And we could see the presence of protein in these animals, and that's in the next slide. No, if we go next. And this is just a histological section of the wild type, untreated, and treated. And in contrast to our editing studies where we would see brown puntate cells, that we do seem to have RNA activity, but not strong enough to achieve reactivation of the gene. So I'll go to the next slide and summarize where we are. So what's next for us? We believe that AAV gene replacement therapy is closest to the clinic of our programs. We're going to leverage the experience with 5 other programs using AAV, ICM, that are enrolling research subjects, so we can draft off this experience. Conduct more monkey studies to confirm safety and determine if gene transfer efficiency we believe is efficient. In parallel, though, we're working with Amicus on a secreted version of UB3A, and if we can demonstrate cross-correction, I think that would leapfrog over our more standard approach, and we're ready to be agile and pivot. We're excited about the microRNA. We think we can use an AAV, but we need more work to achieve more efficient and stable reduction of UB3A ATS. In genome editing, worked much better than I thought it would, but we believe we need a different delivery system other than AAV, and we're going to announce early next week a major collaboration with another faculty member here at Penn who's an expert in LNP mRNA, and we're working on developing that for CNS delivery. So I'll conclude on the last slide where I kind of started, and that is the goalie analogy, that really we want to refine where to take the shot, how we're going to take the shot. Maybe we will decide to take 2 shots, but when we take the shot, I doubt we're going to take it in a way in which we'll have no goalie. But when we do take the shot, we want to make it past the goalie and into the net. So again, thank you, Allison, and thank you all the community for your support over the years. I guess you could say we're on it ×4, but I think it is worthwhile to pursue these in parallel. We don't care what works as long as one of them does. So thank you.
I think we have time for just a couple questions, but I hope everyone appreciates why we recruited him so hard to work for our kids, because we're not here to take one shot on goal. We're all here to ensure that we leverage all opportunities to figure out which is the best shot to take. So thank you, Dr. Wilson.
Yeah, hi. I understand how the gene replacement works in a patient with a full deletion, but how does it work, or indeed can it work in a patient who has a mutation, who obviously has the gene and some form of expression?
Yeah, it's a good question. So the gene replacement is the introduction of a new functioning gene that can cohabitate with an abnormal gene, whether that gene expresses a defective protein or not. It's in addition to.
Sorry, I had a similar question, but about UPD, how would it work?
You know, that's something that we need to think a little bit more about, and I'll have to get back to you on that one.
And we can also answer that in the panel discussion. We have some geneticists in the room that can talk a little bit more about the differences of the approaches for UPD and the different genotypes.